panel and an intergovernmental panel. She's currently the founder and director of the largest community citizen science program focused on pollinators in the world, the Great Sunflower Project. So if you are interested in doing some research on bees yourself, you can go on her website and find out about the Sunflower Project. We were talking ahead of time. I thought it was only sunflowers, but she has now expanded it to all pollinators that may be in your garden. So I encourage you to check that out. So it's with great pleasure that I turn the meeting over to Dr. Gretchen LeBuhn.
Well, thank you so much. I'm so excited to be talking to California Native Plant Society in Marin. I live in Marin, and it's one of the first things that made me feel like I was establishing a home here was back when I used to have time before I had children, was actually coming to CNPS meetings over at the Redwoods, actually. And I learned a lot about the plants of Marin by just listening to talks like the one I'm going to give today. I'm going to share my screen and let's see. Today I'm going to talk about the biology of solitary bees in the Bay Area and really talk about a number of different projects that have been going on done by me and my students and mostly funded by ONNT. I'm going to do like super geeky science stuff here. So please write a question in the chat if something I say doesn't make sense. I've asked my other moderators to interrupt me, and I feel, I'm a teacher, so I'm used to that. So please feel free to do that, and I'll pause a couple times to check in and make sure that we're all together. And I've broken the talk down into 3 different sections. I'll get to that in a minute. And after I've done 2, I'll pause and see if your brains are exploding and it's time for dessert or a cocktail or something to recover from all of this. But let me move forward and get started. I always start my talks about bees, and let me get this a little better situated so you can see my screen share better. Sorry, I'm not quite sure why it didn't fit the first time. With this picture, this is someone who is a carbohydrate avoider's hell. But this is what grocery stores in the Bay Area would look like if we lost pollinators. Our food sources would be primarily wind pollinated, which means grains and grasses, plants that don't need pollinators. And the thing that pollinators bring to us is this. They bring I like to think of it as the sort of lively stuff that have most of our nutrients, most of our excitement for eating, and the things that really improve our diet. So pollinators are really central to food production around the world. And I know you guys are native plant people, but I also always like to start with sort of the flow chart of what pollinators do and sort of how plants and pollinators get put together. So if we start with seeds and fruits, the number of seeds and fruits that are produced by a plant depends on the number of visits that plant gets by pollinators. And it turns out it's the number of visits that a plant receives is a function of both the number of species of pollinators in a habitat and the abundance of those pollinators. And abundance basically is the count of the number of individuals. And I like to say this in part because I gave a talk to a bunch of 3rd graders and asked them what bees did, like what did bees do for plants? And they all, shouted out pollination. And I said, well, what does pollination mean? And they said, well, it means that plants need fertilization. And so bees do that. And so this small child who was actually my daughter raised her hand and said, the bee carries the pollen over and it sprinkles it around the plant to fertilize it, not understanding that it actually was leading to the production of seeds and fruits, not just an increase in nutrients around the plant. So now that we've got the basics done, here's where we're going. I first want to talk about some work that we've done looking at Marin County parks and broom removal. And then I'm going to move on and talk about a monitoring program that we've set up, funded by ONNT to really look at pollinators in the wild areas of the North Bay. And I'm going to report on what we've done on Marin Water District land and state parks land. And then if you haven't pooped out at that point, I'll talk about Napa and Sonoma and what the impacts of fire are on bee communities, and some historical and some look at what's been happening over time. All right, here we go. So I'm a teacher. I like to start with a quiz. If I told you that there were probably over 30,000 species of bees in the world, how many species of bees do you think there are in the United States? You can type that in the chat. Oh, I've got some good numbers here. In the United States, we probably have about 4,000 species of bees. You guys thought we were doing even better than we are. So, which is good, which is larger than almost any other country. It actually is larger than any other country on Earth. And the center of diversity of bees is thought to be in the United States, though I won't be surprised if it ends up being Brazil. Now, if we think about California, there are 1,500 species of native bees in California, and only one species of bees is a honeybee. So that means that there are over 1,499 species of native bees here in California, and one non-native bee, which is the honeybee. There actually, I think, are probably about 5 non-native bees, but the only one that's in real abundance is actually the honeybee. And if we think about Marin County, there are probably over 300 different species of native bees here. We have an amazingly diverse number of bees. All right, so let's go and talk about Marin County parks and broom removal. As most of you know, removing broom is a regular occurrence in any of the lands that are being managed around the Bay Area. It's a major goal of many wildland managers in the Western United States. And we know that invasive plants can actually disrupt plant-pollinator relationships of native plants. And they've been shown to reduce the visitation rate, the number of visits that native plants can get. But there are examples where invasive plants have had what we call a facilitative effect on native and exotic co-flowering plants. So just imagine if you are a small, a native plant flowering next to a broom that has 10,000 flowers, there's the possibility that a bee coming in will veer off to the broom because of the huge amount of nectar and pollen and flowers available there, rather than pollinating a native plant. So given the scale of broom and the amount of effort going into its removal, we thought it was important to understand its impacts on native and non-native plants, particularly on the visitation by pollinators to those plants, and whether or not it had an impact on seed production and fruit production, so that when we're doing removals like this, we understand what the impact is going to be on the rest of the pollinator community. So I did this work with Nevin Cullen. Nevin was a graduate of the formerly Drake High School here in Marin. And he came and worked in my lab at San Francisco State after doing his undergraduate degree at Humboldt. And he's now finishing up a PhD at the University of Pittsburgh, working on some really interesting work on serpentine plants. But Nevin and I went out and set up samples in a series of areas primarily owned by Marin County Parks. So we had 12 sites. You can see it ranges from the Tiburon uplands here to White Hill to Loma Alta, a bunch of parks, as you can see here. Each red dot is a park. And we focused explicitly on the removal of broom because we knew it had a negative impact on plant diversity, and it's such a high priority target for management action. So to assess the effect of removals on pollination services, we first went out and looked at what the background rate of pollination service was. And we used something called a before-after control impact design. And let me just talk you through this because I'm going to show you a second before-after control impact design when we talk about fire. In science, a design like this is considered to be the highest standard because it really gives you the ability to detect the effect of a treatment. So imagine each of these boxes is one of our parks, and all of the parks had broom when we went out there. So that's why they're yellow. So we were out there in 2017. We sampled plants in all of these parks. We counted the number of visits that the plants got. We looked at how many seeds they produced. We looked at the weight of seeds. We looked at what the visitors were to the plants. And so we had a measure of what was happening in 2017. We went back out in 2018 and did exactly the same thing. Well, you just have to look at this year to realize that it's possible that 2018 is a very different year than 2017. So by having this control treatment here, where we can look at what happened from year to year if you didn't change anything, that means we can take into account the shift that just happened due to time and isolate the effect of the impact. It sort of gives us a standard to compare our treatment against. So for half of our sites between 2017 and 2018, there were big broom removal experiments done. The other half of the sites, nothing changed. And so we use that comparison to really sort of solidly look at the effect of the broom treatment. We decided to look at the impact both on non-native plants. These were the 5 non-natives that we focused on, and on native plants. And these were the native plants that we focused on. So we did observations of visits to these. We collected fruits from each of them, and we counted the number of seeds that we collected. And let me start just simply by showing you what we found out about bee visits. And so on this colorful slide, you can see that there's a column for before, and there's a column for after, and we have our control and impact. So if you look at this before column, you can see that the numbers of the control and the numbers of the impact are about the same. This is the total number of visits that plants got to by pollinators. And it should be about the same. There was no treatment that happened, and we were expecting that sites would be really similar. Where it gets interesting is when you look at after. So in this yellow, in the control site, you can see that there was a real decrease in the number of visits that plants got in that second year. If I remember correctly, it was a much drier year than the year before, and we think that's why there was just this decline in visitation. But in the sites where the broom was removed, we saw that the number of visits stayed about the same. And so what this really means is that there was actually a much higher visitation rate than there would have been if the broom had not been removed. So removing broom has a positive effect on those plants or on visitation to plants. Let's see, removing broom has a positive effect on visitation to plants. Now, if we look a little deeper, and I'm going to, I'll first show you the bee visit data in a similar way, and then I'll move on and show you the fruits and seeds. We can actually see a really interesting phenomenon. So I've just graphed those data you saw on that table before here in a different way. So here, the befores on the left, the afters on the right, and the control where there was no change, we did not remove broom, is in red, and the broom removal is in blue. So you can see here's the decline that we saw before, and here's the increase in impact. We decided to look at whether it mattered whether the plants that we were looking at flowered at the same time of the broom or flowered after the broom. And it turned out that we saw a much bigger effect of broom removal on those plants that flowered at the same time. So clearly the broom was actually pulling away pollinators from the plants that were the rest of the plants in the habitat. The interesting thing was when we looked at whether or not there was a difference in fruit production, we actually didn't see any difference. And you can tell that because these two lines are really parallel. And these up and down lines represent the mean. And this is the amount of variation around the mean. And you can see that they overlap. If these two bars were not as overlapping, they would be significantly different. We also saw very little difference. We didn't see a big difference in seed count and seed weight. We have seed count on the top here and seed weight, which are both metrics of fitness. We saw a general decrease in seed count and seed weight, but not a significant one. So just to summarize, by simply going out and looking at plants before and after broom removal, we were able to document what the effects of these treatments were. We saw increased bee visitation frequency to plants that flower at the same time as broom. Removing broom didn't affect a plant's likelihood to produce fruit. We thought it actually might increase seed and fruit production in native plants, but it didn't. We saw a slight reduction actually in the number of seeds produced by plants that flowered after broom. And we saw a slight reduction in the weight of seeds. So this said to us that it's really a good idea to remove broom. There's a clear benefit to pollination of the rest of the plants in the neighborhood. And we expect that these funny results that we had with fruits and seeds will equilibrate over time, and that broom removal is really a good idea. Okay, part one done. Are you still with me? Okay, so let's move on and talk about the work that we've been doing monitoring bees on Mount Tamalpais, and this was all funded by ONNT. So so we hope ONNT comes back and really does continues their excellent work. So some of you may have participated in the evaluation of what was known about Mount Tamalpais that happened, I guess, about 5 years ago now. When the first report on Mount Tamalpais natural resources came out, it became really clear that there was a big gap concerning the knowledge about insects. Paul De Silva said only the roughest estimates about insect diversity can be made in absence of baseline information from a detailed insect survey. And given Mount Tam's complex and varied topography, geology, vegetation, communities, and microclimates, some experts suggest that the number of insects could be 6 to 10 times greater than the number of plants found on the mountain. And I think this is really true. So So I think this is really true. The first step that that the group at ONNTTAM decided to take on was to look at pollinators, because they're indicators both of species richness, the number of species, and of the ecosystem service of pollination being provided by those pollinators. Okay, so I want to start with the end. There are a couple key points that I'm going to make in this part of the talk, and I think these are the real take-homes from the monitoring that we're doing. The first one is that the Marin Water District and Mount Tam State Park sites really support a high diversity of bee species, and they have several that are of special interest. We did not find evidence that there's been a loss of some rare species, of some of the rare species of plants is having a significant impact on pollinator species. That was something that was of concern to the group. And then our analysis suggests that the fuel reduction treatments that are being tested up on Mount Tam need to be looked at carefully, because we had, well, I'll show you, we have some evidence that there may be a significant impact on the abundance of bees. And then I want to draw your attention to notice that there's a really high diversity of species in some of the coastal chaparral sites that we sampled. And there are very few studies in these habitats, and this high diversity was really unexpected and exciting to see. We are continuing. This year, we're doing a lot of work in coastal chaparral in the Marin Headlands and up on Point Reyes. So we hope to really expand what's known about that. So when we were hired to work on Mount Tam, we were given a set of goals. The first one was to gather what historical data was known on pollinator taxa of Marin, which meant going to all of the museums around the Bay Area and going through the insect collections to see which of these sort of what had been collected over time. The second one was to look at whether or not there was any risk to pollinators of plant populations that were in severe decline. And then a third one was to create a species list from current and historical data. So we went out to try and gather as many bees as we could in a single year to supplement what was known from historical data. And then we're starting to put together distribution maps for different species and to give some management guidance for fuels and for invasive species. So the San Francisco Bay Area is really one of the most biodiverse regions of California, and it supports a small but thriving agricultural community. So pollinators are really key. What we're really reporting on is sort of a baseline for monitoring the pollinators as we go forward. So we went out and sampled 20 sites, 4 state park sites and 16 sites in the Water District, all on Mount Tam, which I think of as the ecological anchor of Marin County. These surveys that we did are really important because they establish a baseline by which both other natural habitats in the region can be compared, but they also will allow us to document change over time if we go back on a 5-year basis and see how things are changing. And I do want to say that the value of this type of survey work really builds over time. And the initial products are things like maps of the distribution, databases of the records, specimens for collections and for people to use for DNA barcoding and understanding taxonomy. But as these sites get resampled in 3 to 5 years, we're really going to be able to detect change in abundance in common species. And as the sampling builds 5, 10, 15, 20 years out, we'll even be able to detect changes in individual species that are not that common. Okay, so what was our approach? Well, for the historical data, as I said, we went to museum collections and we gathered we got all the records we could from Marin County. We said Marin County because so many of the historical records will say something like Fairfax, and you don't know whether it was on Mount Tam or not. And so by focusing on the county and treating that as an entity, we felt like that was a more valid way to collect data. And then for the pollinators of rare plants, we went to the scientific literature and we looked for every publication that recorded visitation of visitation to that plant or recorded that plant as being important for that pollinator. When there were not records that were specific to that plant, we looked for closely related plant species and at least drew in that information as potential pollinators. And then for the survey work, as I said, we sampled 20 sites and we decided to embed an experiment in those data so that we could maximize the information coming out. We did both the forest treatment. We both embedded that forest treatment experiment into the water district land. So we had half our sites in forest treatment plots, half our sites not. But we also were interested in whether or not the bees in the interior of Mount Tam were different than what you found in the wildland urban interface. So is there an impact of the residential habitat around the mountain that is influencing the bee communities? And in particular, you might expect if there's a large effect of honeybees or a large number of honeybees in those areas, they may have an effect on the native pollinators. And then we had sort of a side interest in what the difference would be between the coastal side of the mountain and the bay side of the mountain. And we haven't done much with that yet, aside from just looking at the abundance, but that's something that will continue. And then I talked about the Marin County broom experiment. Sorry, I forgot that I meant to put that first. All right, so let's go. Here are some of our results. We looked at historical records from Berkeley, Davis, Riverside, Cal Academy, USDA, American Museum of Natural History, and every online data repository that we could find. And we were able to find records of 38 genera and 242 species having been recorded from Marin County. The largest number of records came from bumblebees, which is this column here, Bombus. And if you look over here on the left, I've just put a list together, a list of what plants these records were found on. And you can see that bumblebees really pollinate a diversity of the genera that are found in our native fauna. So they're really important generalist pollinators. When we think about the pollinators of rare plants, there are 211 rare plant species on Mount Tam. We looked at each of those species and identified what we thought the pollinators would be. There were 48 species that were clearly not animal pollinated, that they were wind pollinated. There were 101. There's no information in the literature. And it would be really interesting to have someone go out and really do observational work trying to document who are the pollinators of these species. Most of them, we were able to find something about the genus, but not something about the species. But for 62 species, we were able to identify their animal pollinators. And these were primarily bee pollinated, and some of them were hummingbird pollinated. And most of them relied on generalist pollinators, particularly bumblebees for pollination. There were also leafcutter bees, Osmia and Megachile, that were pretty common, but those were the dominant. When we did this, we actually got pretty detailed data. So here's an example of what the data looks like. This is the species Rhododendron macrophyllum. I actually don't know its common name. It's California Rose Bay. We looked at what pollinators were in Marin that had been recorded on it and found 2 species of bumblebee. And then we looked at are those pollinators found outside of Marin, outside in other parts of California? Are those pollinators found outside of outside of California? And you can see that for this species, there are records from other parts of California that it's also pollinated by the Bombus flavifrons. So this is a good example of a species that neither of these pollinators are rare in our area. So the reduction in this area of this plant should not have a big impact on any of these pollinators listed here. Okay, so let's talk about the really fun stuff where we looked at the full bee community. Just to remind you, we had 20 sites, but you can parcel them out in a number of different ways. There were 4 coastal sites. There were 8 sites that we called interior, so they were not in the wildland urban interface of Mount Tamalpais. There were 8 sites that were in the wildland urban interface. There were 7 sites in forest treatments. So this picture here on the left is a forest treatment where the understory has been cleared out to reduce the fuel load. And there were 9 sites that were in control where that understory had not been removed. At each of these sites, we did field surveys of bees. We collected all of the pollinators in traps. This is the distribution of the sites, and you can see the colors as coded. There's color coding here to tell you how we classified each of these sites. And I'm just going to pause for a minute so you can look at this in case you're someone who's really familiar with these areas. If you're looking for places to look at wildflowers, particularly in the spring, these are some of the most wonderful places to visit in the Water District and on state park land. All right. So what did we found? In aggregate at these sites, we found 31 genera and 122 different species. 28% of the species we found, 42 of the bee species, had not been previously documented as being from Marin County. So this brings the cumulative total of species detected in Marin to about 280. Of the historically recorded species in Marin, we collected about 40%. And that's not unexpected. We were collecting primarily in grassland. We did not do extensive work in chaparral. And there's a lot of variation from year to year. And I expect that over time, as sites get resampled, we will build towards this maximum, or towards a number up around 300 species as we continue. We had some really interesting bees, and I want to show you some pictures. So this is a bee species, Ashmeadiella cactorum. It's rare. This species is really uncommon in coastal California, including in the Coast Ranges. And there's a single historic record from Marin County, collected in 1955 by Paul Hurd, who was at Berkeley, and Charles Michener, who was at Kansas. And there's one record of this species from Santa Clara and one from Napa. This is a generalist species. It visits lots of different plants, but it was really exciting to get one of these. We found an exotic bee species, Anthidium manicatum. Interestingly, there are only a few records of this bee from California, and most of them are from coastal central California. There are some records of this species from the East Bay and Discover Life. I actually have seen this in my garden, so I knew it was in Marin, but it had not been formally collected, and therefore there was no scientific record that it was here. This was the first time it had been recorded from Marin County, though it probably, or I know at least that it had arrived here earlier. There was a bee we were hoping to find that we did not get. It actually looks a lot like that, that Anthidium that you saw earlier. This is Trachusa gummifera. This species is only known from Marin County and from San Francisco County. It was collected once up at Carson Ridge. A couple different of these were collected up there. And there's a single specimen that was collected in 1957 in San Francisco. It's thought to be extinct in San Francisco. My lab's done quite a bit of work in San Francisco and we haven't detected it. But I was hoping we would pick this up. And I would like to go back and actually really spend some time on Carson Ridge to see if I can locate where this bee species might be. All right, so those are some of the players that we found. Let me show you some of the differences and information we found about the different areas. So I want to start by talking about site differences between coastal and interior areas. The first thing I want to draw your attention to is the abundance of bees. So this is the total number of individuals that we caught. And you can see these graphs, this line here, mean, so the average. The box here tells you the amount of variation. And when there's overlap like this, there's not a significant difference. But you can see that there are trends in different ways. So the first thing you can see here is that the coastal sites were very similar to each other. These boxes aren't very big relative to the wildland urban interface, which had a wide range of abundance of bees there. The interior areas were in between. If we look at the number of species, you can see that the coastal sites actually had slightly higher average number of species than the interior and the WUI, but they were pretty similar. So the species richness, which is the number of species, was higher at the coastal sites, and abundance was higher at the wildland urban sites. And so in ecology, we talk a lot about diversity. And when you have a lot of species, when you collect very few individuals, we consider a site to be very diverse relative to a site where you have a lot of species, but you caught a lot of individuals. So that really makes us think that those coastal sites are really, really important. The abundance in these sites was largely driven by a single species, Halictus tripartitus. And this is an incredibly common bee throughout all of Central California. And it's almost always 50% of the abundance of bees that we find in pan traps. And that's partly a function of they love pan traps, and it's partly a function that they are actually really that common in these areas. They're like the workhorse bee of Central California. Let's move on and look at the fuels management treatments. So the water district does fuels management to try and reduce the fuel load. And they've been experimenting with some different ways to decrease the understory. And for bees, we weren't sure what this would mean for bee populations. When they go in and do these fuel management treatments, there's quite a bit of disturbance of the ground. There's the removal of cane or of potential nesting sites, but it also really opens up that understory and lets light in. And bees do not like heavy forest cover. They avoid shade. So we weren't sure whether the fuel reduction treatments would lead to an increase or decrease in bee use of these habitats. Okay, so let me orient the interior sites on the right. And the two different colors are the controls, which are in red, which are sites that did not receive a fuel reduction treatment, and the fuels, which are the sites that did receive a fuel reduction treatment. So you can see here that in both the interior areas and in the WUI, there was a reduction in the number of bee species in the areas that had the fuel treatments. So we're not sure what's driving this, but there's clearly some sort of impact. One of the reasons, so I showed you the before after control impact design before, and I'd be more comfortable drawing conclusions if we knew that these forest treatment plots had the same number of species prior to the fuel treatments going in. So we don't know whether the places that were chosen for the fuel treatments happen to be slightly different than these controlled plots. And so this is an artifact of they needed fuel treatments because they were a certain way, or whether this is really because of the fuel treatment. And so our recommendation to the water district is that, you know, they want to pay attention to this as they continue to do fuel treatments. We also saw shifts in abundance of individuals. The previous slide was the number of species. And interestingly, we saw a reduction, once again, in the abundance of individuals in the interior. But in the WUI, the wildland urban interface, we actually saw that the fuel reduction treatment led to a big increase in abundance. I haven't gone back to look at what caused this increase in abundance. Was it primarily those Halictus tripartitis? But it also might actually be something like an increase in honeybees in that area. So that's something that we'd like to go back and look at more closely. Okay, so to summarize what we saw with the fuels management, in the interior sites, the fuels management sites really seem to merit some more attention because there seemed to be declines in richness and abundance. And then we saw that increase in abundance in the WUI sites that is worth pursuing, because not all increases in abundance are positive. So it might mean that there's one species that's really taking over in that area, and we'd just like to know a little bit more about what that is. All right, so in our work with the water district, I want to bring you back to sort of what are the key takeaways from this are. The first one is that we saw a really high diversity of bee species. It was really exciting to look at what we caught. And we had a bunch of species of special interest. We didn't find evidence that the loss of some of the rare plant species is impacting the pollinator species, which was a relief. And then the analysis suggests that a really careful look needs to be taken at the fuels reduction treatments, because there does seem to be an impact on bee populations there. And then I showed you that there's a high diversity of species relative to the abundance that we were getting in the coastal chaparral sites. And this is one of the first studies done in those areas. And so this was unexpected and exciting to see. Let me wrap up talking about monitoring by just giving you a vision for where it's going to go in the future. Right now, there are a bunch of other sort of products that come out of this work. The first one is that there's now a public database of all of the records of bees in Marin County that resides at the USDA, which houses the National, oh, they keep changing their name, National Pollinator Monitoring something or rather. They have a museum of all of the bees of the world there. And they maintain a database of all of the records of bees collected in the United States. And these data are now there and available. We collected tons of specimens that are being used and distributed to other collections, so people will know what the species look like. There is DNA work going on where people are looking at the genes in these bees, and they will be used for revising taxonomy and are starting to be used for public education and display. You should really, pretty soon, I think you'll start seeing photographs, really beautiful macro lens photographs of the bees that we collected, and they're just spectacular. In 5 years, when these sites, actually in 3 years, when these sites get resurveyed, that's when we start getting some even more exciting results. We'll start to get maps of the distributions of the different species. We'll start to see if those distributions are changing. So on the right here, you can see some data from a bee species that's collected by citizen scientists in Great Britain. And from 1900 to 1950, you can see that this was a bumblebee, was pretty well distributed across most of England below Scotland, and that by 2000, 2007, they were only found in the southern part of England. So there was a dramatic change in the distribution of this bee species. And that's the kind of thing we'll be able to start to see here in Marin. For the overall group of pollinators, we'll see changes in abundance, changes in total species. For the genera and what we call major guilds, these are things like cavity nesting bees or ground nesting bees or short-tongued bees. We'll start to be able to document changes in those and changes in abundant species. And then over time, as we get out to 10 years, we'll really be able to look at trends for individual species and detect large crashes for any given year. So this will give us that early warning signal that we need to intervene to make sure that we're keeping a healthy habitat. And it'll start to allow us to compare the magnitude of changes that we're experiencing here in Marin with other regions where people are doing this type of monitoring. So I applaud the ONNTTAM group for recognizing the importance of establishing baselines and doing monitoring of insects like this. I think it's really important for maintaining health. I'm going to pause there. Maybe I'll answer a couple. Whether you have some stamina and want to hear a little bit more about fire, or whether this is a good spot to break up. I'm also going to turn on the light because it's gone dark in my room, as you can see here. And great. That's a little better.
Fire is on everybody's mind. So I think I would say, go ahead.
All right.
And
if you drop off now, I don't take offense. It's been an hour. And I think that I know from teaching undergraduates that 40 minutes is a long time. So please go forth and do something if that's next. All right. Here we go. Napa Sonoma and fire. In the early 2000s, I went out and did a study of how vineyard expansion into oak woodland in Napa and Sonoma reshaped bee communities and reshaped plant communities and the structure of their interactions. And I was lucky enough in 2017, was it 2017? Yes, in 2017, to have an amazing graduate student, Molly Martin, join my lab. And I guess it was, can that be right? Was it, she came in 2016 because it was fall of 2016 and the fires were in fall of 2017. Over that year, we planned, actually, no, she came in 2017. Over that year, we thought, wow, wouldn't it be neat to go back out and restamp these sites? They had not burned point because we were interested in whether or not there was a change over time, because things have happened in the 15 years since I went out and did the early 2000 work. Well, we got all our permissions and we started working on this, and lo and behold, there were the tragic fires of 2017. And the fires ended up burning almost exactly half the sites from the original study, which made my little scientist's heart go pitter-pat, because that's setting up almost a perfect before-after control impact design. Though, it was just awful what happened there. It gave us the ability to look at the impact of fire while controlling for the change that happened over time. Here's a map of where we were. Our study sites were distributed throughout Napa and Sonoma counties, or really Napa Valley and Sonoma Valley. And we had 4 burn sites, which you can see here in red. And we had 4 unburned sites, which you can see here in green. And just to orient you, this is the town of Napa here. This is Deer Park up here. Here is Temelec. This is Sonoma Valley here. And so you can see we had 3 sites, part of this fire, 1 site over here, and a total of 8 sites. All of these sites were categorized as lightly burned. They had really similar burn profiles. And what this set up for us was a before-after control impact design. The earlier data was from 2002 and 2000C. So we had 8 sites we had data from 8 sites in 2002, 8 sites in 2003, and then we collected new data from 4 sites that had burned and 4 sites that had not. Now, most studies that have looked at the impact of disturbance compare sites that were impacted to ones that weren't. But being able to sample the same sites is really the strongest study design for detecting change. So this was really an exciting way to be able to get a sense of what was going on. So let me give you just a visual so you have some sense of the sites that we worked at. Most of the sites were varied from this being our most dense oak woodland to a relatively open field like this one. They were some sites were pretty near vineyards, and some were in more natural oak woodland habitat. And the fire severity varied a little bit across sites, but not as much as you would think. We went out and hand netted each of these sites using the same protocol that we had used from the original study. We visited each site every 2 weeks and sampled for 2 hours. And we would walk through a site and catch every bee that we saw. We'd spend no more than 5 minutes at any particular plant. So we would make sure we had walked through the whole area. And we only sampled on warm, calm days so that we could try to minimize the effects due to winter. We collected each bee. Here's a Bombus vosgesensky, and then pinned it and labeled it with the information so that it could be, so that we have a record of what was there. Bumblebees, we did not always collect because we can identify many of those on the wing. So we usually only took a bumblebee if we needed a record from that particular site. When it came to analyzing the data, the first thing we wanted to make sure that we could eliminate as a factor was the idea that there were big differences in weather. And so the blue bars here are the bars from the year of the fire. And you can see that there's not a huge difference in minimum temperature by month or maximum temperature by month, or even precipitation. 2017, 2018 was slightly cooler and slightly drier, but not enough that you would expect it to have a significant impact on the bees and the plants. Okay, so let's see what happened. Oh, let me get rid of this, sorry. So when we compared burned versus unburned sites for the total abundance of bees, let me walk you through sort of how these graphs. On the left here is a graph that shows you what we expected to see. The left graph shows the 4 control sites. The right graph shows the 4 burned sites. Within each graph, the left dots are the before and the right dots are the after. The individual lines show the change at each site, and the variability in each site. Those things are due to differences in management and differences in habitat. What I really want you to pay attention to are these boxes here. These boxes are called box plots. And the line in the middle once again, is the mean value. And the bottom of the box is the first quartile, so the first 25%. And the top of the box is the third quartile, so the 75%. So by looking at these boxes, you can see sort of where the average was and the amount of variability at each site. The first thing I usually look at, and what you should look at, is sort of how the means change in the expected sample, and then go look at what happened when we had real data. So we expected to see higher bee abundance in burned areas relative to unburned areas. And this is mostly because fire creates conditions where annual plant species that are easily accessible and loved do best. While there's a significant difference in bee abundance between the burned and unburned sites, it really looks like there were major declines across both types of site between the before data and the after data, and not much difference due to fire. So here's the before at the control, here's after, there were huge declines. Here's the before in fire, and here's the after in fire. When you look at the numbers for this, and you plot the bee abundance at all sites on a single graph, you can really see there were significant declines in bee abundance. We separated out 2002 and 2003 versus 2018. We found huge declines. We found a 67% decline if you use 2002 as the baseline, and a 50% decline if you use 2003 as the baseline. And so what this says to me is that we need to be paying attention to our bee communities. Some of these sites were in wildflower preserves. Many of them are in sites in areas that really get no disturbance. And it suggests that there may be some regional declines in bee populations that are important to detect. And that's one reason that I'm so glad that we've started the sampling up on Mount Tam. If we look at bee species richness, so richness is the number of species. I'm just getting rid of that. I'm sorry, I thought I could do this here. Since I'm not projecting it, you see that little thing. So bee richness is a measure of the difference of species found at each site. And this, just tells us about the number, but it doesn't mean the same species are present at different sites. So look at this graph on the left, our expected, and compare it to what happened on the right. Similarly to bee abundance, we expected to see an increase in bee species richness for the same reason that we expected there to be a flourishing of annual plants. But we didn't see that increase in bee species richness. We saw significant declines in the number of species that were detected. And if you put some numbers to those declines, it was actually a 16% decline in the number of species, and if you compare it to 2002, and a 32% decline from 2003. The last thing I want to show you is sort of a graph that shows you how the bee communities move in space. So you can go home and tell your favorite person who loves statistics that you understand non-metric multi-dimensional scaling after this, and I'm going to take you there. All right, so to look at communities, what we tend to do in science is use something called non-metric multi-dimensional scaling. I'm just going to call it MMDS, so I won't trip over it. This is a technique used to represent communities in multi-dimensional space. So if you look at this first line here, you can plot the abundance of one species on this line. Here I've got community C didn't have any, community B had 3, community A had 5. Now, if you wanted to add a second species to say something about the community, you could do it by graphing it like this, where we have two dimensions. We have the abundance of species 1 here, we have the abundance of species 2. So we have community A, community B, and community C. And you can see that they're different, like the bees are different, though they're, community C have similar numbers of species too. they all have different numbers of species species 1, which we knew from the first one. And so what NMDS does this in multidimensional space. Here's an example of what it looks like for 3. We've added a 3rd species here. And you can see, you can really start to see how communities are different. What an NMDS does is basically squishes all these axes together and then picks the 2 most representative measures and creates a plot based on those. So here are the plots for the 2002 to 2017 data, and this should say, sorry, 2003 to 2017 data. Each of these dots represents one of the years at one of the sites, and they're color coded. So you can see before, we had our before control sites and our before burn sites. They overlap, as you might expect, because they were all similar randomly chosen sites in Napa and Sonoma. And you can see that after burning, the burn site community becomes different. So while we weren't able to detect differences in the number of bee species and abundance, the composition of the community really changed. We also saw differences in the control sites before and after. And I think that also reflects the change that we're seeing over time that has me concerned. If you look at we see the same pattern with 2003. There's overlap in the before burned and before control. There are differences between the after burned and after control, and there's a big shift that's happening over time. So what this really means is that over the 15 years between when we sampled originally, the species changed a lot. It also means that the burn sites were made up of largely different bee species than unburned sites. And the next analysis that we want to do that we haven't done yet is really to look at what families and genera of bees are being most affected by the changes that we're seeing over time. If I think about what might be driving this change, one of the things that we thought might be doing it is what's happening with the plant community. So I'm going to show you the data presented in the same way for what we saw for plant species richness and for native plants. So these are data collected at exactly the same sites. This was our expectation for plants. As I said before, we thought that plants would increase. Like bee abundance and species richness, we thought we'd see an increase after fire because of the increase in available nutrients, the increased niche space. And gosh, you guys all know that there are fire followers that are only found in the years following fire. But in fact, we saw declines in plant species richness at almost all of these sites. There were 2 sites that we didn't, but overall, there was a big decline in species richness across both the control and impact sites. If we take that down a level further and we look at native plants, we didn't actually expect to find much change in native plant species, but we actually saw a decrease in native plants at all but one site. And so you can see that both the control sites and the burn sites really experienced declines in native plant species, which means that we were seeing also an increase in non-native plant species. If we look at the percent invasives, we didn't expect to see a big change in the percent of invasives, but we found that invasives increased both across burned and unburned sites over time. There was not a significant difference between the burned and the unburned sites, but something's happening up in the North Bay that's seeing significant shifts in the plant communities. So let me just summarize this. We saw bee abundance decline 67%. The overall bee numbers, the number of bee species really dropped dramatically over the past 15 to 16 years. And it may be even so overpowering that it's drowning out some of the changes we might have seen it following a disturbance like wildfire. These declines are really similar to the insect declines that have been detected around the world. And similarly to some of these other declines, it's really surprising considering that they're occurring in areas that aren't experiencing a lot of landscape change. I mean, really, if you think about Sonoma Valley and Napa Valley, there's not a ton of change that's happened in the last 15 years. There is there's been an increase in the use of neonicotinoid pesticides in some areas, but I was really surprised by this. One other possibility might be that, we had a significant drought in the time between 2002 and 2015. And so, that is another factor that might be driving some of these changes. I think I'll stop there. I do want to leave you with a couple key messages from this last part. The first one being that these overall bee numbers are something we should be paying attention to. But I do want to say that I was really heartened to see how resilient the bees were to fire. And it did make me feel much more comfortable that these big wildfires that we are having are not the thing that needs to be of most concern for maintaining our bee and plant populations. I'm more concerned about either the impact of drought or the impact of some of the pesticides. I'd like to know more about what's happening there. The dark side of me, which is not heartened by the signs of resilience that we see in this work, is that these massive declines really could create a negative feedback loop, whereby if we're getting these declines in pollinators, it's feeding back into declines in seed production that could lead to declines in native plants, that could lead to declines in pollinators. So we've gone back and done a little more work in Napa and Sonoma, and we're now working up in Point Reyes, and we're hoping to disentangle this a little bit more. And we've collected another year of data, the year after these bee declines that we're in the process of analyzing, to just make sure that there wasn't something in our year that caused us to detect this magnitude of change. I do want to not just end on a complete bummer. I do think we have the knowledge and tools necessary to slow, if not reverse, these declines. But it may take large-scale coordination between scientists and policymakers and land managers and organizations like ONNTTAM and people like you who pay attention to our native plants and can really start to build the foundation upon which these pollinators rely. All right, now I'll say thank you and be done.
Okay, I think I'll take the questions first that are in the chat. There are not that many of them.
Okay.
This is a question that goes back to the Mount Tam monitoring, and from Paget Lenars. Graphs show apparently significant effect on seeds, but your lecture said not. Please clarify.
So yes, there is a significant effect of removal of broom. There's a decline in the number of seeds that are produced. It's a small effect, and I don't quite know what to do about it, given that we saw increases in pollinator visitation rates. There are two reasons that you might expect that an increase in pollinator visitation could cause a decrease in seed production. One is that maybe bees are bringing in pollen from other plants. So before they came to that plant, they were at another not heterospecific, so not plant of a different species, and they deposited the wrong pollen on. There's also well, that would be the main reason. There's another other reason that I thought of that could happen, that at 8:44 without dinner has gone from my brain. Yes, you were right. There was a significant decline because it doesn't follow on the visitation rate. I'm not quite sure what to do about that.
Jane Aney asks, what is the difference between the interior and the wild and the WUI sites?
So let me actually pull the map back up. Hold on, I think I can get to that pretty quickly, because it's easier to see if I show the map. So if you look here, all of these sites in the green are what we call the wildland urban interface. So these are sites that are within a particular, within, I think we used a 5-mile radius, have a certain proportion of that land is residential. I think we did some GIS analysis that showed that, how much built area there was within 5 miles of that area. And you can see less so up here. These were the ones that were closest to being interior. But like these sites here clearly are going right up against Fairfax and Kentfield and Mill Valley. Whereas these sites here are much further from those urban areas. And so these would be interior sites and these would be wildland urban interface sites.
Okay. Bob Maselli asks, how are you catching the bees in your population and monitoring work?
So we use stadium beer cups that we set out. We put 9 at each site, so we feel comfortable that we're not oversampling. And we put a preservative called propylene glycol mixed with water in the cups. The propylene glycol sits at the bottom, and so bees land on these cups and fall to the bottom and are preserved. And then we clean them up and make them fluffy again and add them to the museum.
Laura Lovett wants to know, how many specimens did you collect in total?
I think that we got, a little over 15,000 specimens across all 20 sites. So this is, and that sounds like a lot. It may be closer to 20. If you think about that's over a 6-month period of time. So on average, we get about 10 to 15 bees each time we go out and pick up the cups. We think that we get about 3 bees per cup per day at the peak time. And we've done some work with some collaborators up in the Rocky Mountains to see whether or not these cups actually have an impact on bee populations. And so in the Rocky Mountains, we put out cups in a set of meadows, and we put no cups in another set of meadows, and tracked for 5 years or collected bees for 5 years in the first set of meadows, but not in the second. And we then compared the number of species and the abundance in bees in the meadows where no collecting had happened to the number of species and abundance of bees in the meadows where there'd been pretty intense sampling. And there was no difference between the number of species, the abundance of bees, which made us all comfortable that this type of collecting is efficient, but not oversampling in these areas.
Annette Anzalone asks, have you studied any areas that have been restored with native plants?
No, we would love to do that. So no, not in Marin. We have done that down in Monterey and looked at dune restoration. And we've been able to show that there's a much higher diversity in the restored dunes than in the unrestored dunes.
Paget Linares asked, do you think that increase in vineyards has had an effect on the bee abundance in your study?
I have not gone back to look at whether the acreage of vineyards changed significantly over that time period in the Napa and Sonoma Valley. My gut feeling from driving around there is that there has been a small increase, but nothing like what we've seen in Southern California or in some of the less well-known valleys of California. We should go look at that. I should have those numbers, in hand. It's a really good point.
Your fire follower study showed a great correlation between reduction in bees and reduction in native plants.
Yeah.
What are your thoughts about the direction of causality in that?
Boy, my intuition is that both of those are related to drought and to a change in climate. I think that I was very surprised that there was a reduction in native plants. I was surprised we saw a reduction in native bees. It was really completely unexpected with all I know about bees, I did not expect to see anywhere near that magnitude of decline. And I lose sleep over it. It really makes me anxious. But I think that it may be an effect of climate change, and in particular that significant drought that we all went through 10 years ago.
But coming down from the ultimate cause to the immediate cause, is it because the bees don't have the nectar and pollen sources they need in native plants, or is it the plants don't have the bee pollinator services they need? Which are both.
I think it's a chicken question. I don't know.
Were there any particular vegetation treatment techniques that were more problematic, or what was the variety of vegetation treatment management strategies that were used by MMWD that you studied that appeared to have a negative impact?
So the only thing that we looked at was the fuels reduction plots. And, clearly Mount Tam needs, they need to manage fuels when there's so many people living close to wild areas. And so it makes sense to clear out the understory and find some ways to reduce the potential fire hazard in these areas.
But is there any difference, for example, between?
I don't know whether the impact, whether there's some ways to. Sorry, could you repeat that?
Yeah, I was just wondering. There are a variety of different vegetation treatment strategies, including, handwork, mastication, controlled burn. And I'm just wondering whether you got into the details of what kind of vegetation treatment strategy they were using and its effects.
At all of these sites, they were done in the same way, and they used mechanical removal. So they had big stuff in there, clearing out the understory. It would be interesting to do a comparison of all of those different methods of doing fuels reduction to see which one is best for, plants, insects, and other animals that use those habitats. But once, doing that type of costs money, it's not easy work. And all of our, agencies are struggling to keep doing survey work like this.
Yeah. Are there any other questions that folks would like to ask individually? If so, I can only see a handful of you. If you could raise your hand through the reaction button.
I see there's a question about climate change. And no, we have not yet looked at climate change. Climate change is easy and hard to study because we really just have one region of California that we've been sampling in that's been experiencing, the same changes in temperature over time. if we had, this site, if we had sites distributed across California where the impacts of climate change are different, we could start to isolate the effects. But I do really think that some of this is related to climate change. We've done some work up in the High Sierra, or up actually in the Central Sierra, looking at bumblebees. And we've done some modeling there where we've shown that that restored meadows should help to offset the impacts of climate change, because they have much healthier hydrology that will maintain plants for longer periods of time. And that's important for maintaining a diversity of bees. But we haven't done anything like that down here in the Bay Area.
Okay, it looks like that is all the questions. I really want to thank you. This is important work, and it's not the most optimistic, but it's important for us to know what's going on. It's for us to not be Pollyanna-ish.
You guys should all give a shout out to ONNTAM, who pulled together the funding for this from a variety of different sources. And I really think that ONNTAM is a revolution for understanding what's happening in Marin County, that they've really done an amazing job of identifying where we have knowledge gaps and trying to find the funding to fill it. And I'm just so grateful to them for funding this work and for really thinking deeply and profoundly about what we need in Marin County to maintain the wonderful biodiversity that's here.
And we certainly hope that you're able to continue this work, because as the more longitudinal studies we can get, the better off we're going to be. And it's great that you're able to have that kind of time perspective. So
yeah, I agree completely.
Thank you so much.
It's really sure. Thank you.
About next month's program is still up in the air. We'll announce that by email and on the website. So stay tuned. Paul De Silva is the head of our scholarship committee, and if Paul, where are you? He wanted to announce the, oh, here you are. He wanted to announce the recent scholarship awards that the chapter gave to some very interesting student research work.
Yes, well, since we were just speaking of funding, it's very appropriate that we remind everyone that the Marin chapter of CNPS has committed itself to encouraging the next generation of researchers. And so each year we have a scholarship competition. And this year, we're very proud to announce that we've awarded 3 scholarships. Emma Fryer of Cal Poly San Luis Obispo has received the Howard Cohn Memorial Scholarship for the proposal entitled Verdict Clay Endemic Desert Annuals of the San Joaquin Desert. Sushmita Podell of UC Santa Cruz is receiving the Special Award of Merit for her proposal entitled Study of Blue Oak Ecotypes across Mesic and Xeric sites in California. And Nicola Bañez of the University of San Francisco will receive a Special Award of Merit for her proposal, Climate as a Driver of Divergence in Soil Specialist Plants. And so we wish all the winners great success in their work and look forward to learning of the results of their research, perhaps at a future meeting. And we should also thank very much, of course, our prime benefactors, Joe Cohn and Ken Howard, who left the first funds as bequests, and they were very strong supporters of the Marin chapter. So thanks very much.
Thank you, Paul. And we're right at 9 o'clock. That's great timing. And again, Dr. LeBuhn, it was a very, very informative talk, and we're so appreciative of you giving us this amount of information. So thank you so much, and we hope to see you all next month.